Okay, take a look at this standard exam answer. RNA polymerase makes mRNA. I mean, you'd think that's a pretty solid, basic biological fact, right? Well, in the Cambridge A-Level Biology 9700 exam, writing down that exact phrase is going to instantly score you a 0. No joke. So today in this explainer, we're uncovering exactly why that seemingly perfect answer guarantees a failing grade and how you can fix it. Let's compare that standard 0-mark answer to a top-band response. Notice the difference? The top answer says RNA polymerase binds to the specific promoter region. Now, that shift in wording isn't just some fancy stylistic choice. It's the literal difference between dropping a great boundary and securing your target score. Examiners are actively hunting for these highly specific phrases, and missing them is a massive, massive risk. So let's dive into this. Why exactly does that generic answer score a devastating 0? I mean, it is biologically true that RNA polymerase is involved in making mRNA, right? So why the 0? Well, the truth is, at the advanced level, examiners aren't just testing your ability to memorize a final biological conclusion. They are testing a completely different cognitive domain. And the secret here lies in what's known as the method mark strategy. The academic benchmarks for this syllabus prioritize analytical depth way over superficial breadth. What does that mean for you? It means marks are awarded sequentially for demonstrating the exact procedural process. You don't just get a free point for knowing the final destination. You actually have to earn those marks by proving you understand the exact biological mechanism and the specific steps taken to get there. So how exactly do we demonstrate this analytical depth on paper? We do it by following a really strict, standardized sequence for literally every transcription question. First up, step 1, identify the key elements from the stimulus, like the specific type of RNA polymerase. Then, step 2, apply the biological principles of transcription. Step 3, execute your analysis with super clear step-by-step working. And finally, step 4, verify your results and format them using the proper terminology. Honestly, this is the exact procedural sequence that unlocks the mark scheme. And if you want to push into those absolute top bands, listen to this quote pulled straight from a principal examiner report. Candidates referencing cross-topic connections achieve significantly higher marks. You see, the theoretical architecture of transcription doesn't just exist in a vacuum. It rests on precise definitions and, crucially, your ability to integrate this specific mRNA formation process with totally different areas of your biology syllabus. Now, in just a moment, I'm going to reveal a specific examiner clue that completely changes how you structure a transcription answer. It's a real game changer. But first, you really need the framework that makes that clue completely obvious. Moving on to Section 1, the analytical framework from concept to wording. This framework is basically how you translate a biological concept in your head into the perfect wording on the exam page. Top-scoring candidates deconstruct every single question into 4 distinct pillars. Column 1 is the concept, meaning the underlying biological fact. Column 2 is the evidence. That's the specific data or context you pull directly from the exam stimulus. Column 3 is the scientific reasoning, which explains the why behind the mechanism. And column 4 is examiner wording, which ensures you're using the exact scientific terminology required. To guarantee full marks, you absolutely need all 4 of these pillars working together. All right, let's actually put this framework to the test. Imagine you're presented with this experimental observation. RNA polymerase binds to DNA, creating an mRNA strand. Just pause for a second and think about it. Can you spot the fatal error here? What is the missing context in this seemingly correct scenario? Now, what's really fascinating about this is how that missing context completely alters the required answer. A generic textbook-style description just isn't going to cut it anymore. You need a highly specific application that links directly back to the provided data stimulus. We're talking about mentioning the specific environmental variables, or maybe the exact mutated promoter region that was literally given to you in the exam paper. And this brilliantly illustrates our next major point, straight from the examiners themselves. The classic mistake is providing generic answers not specific to the question context. Always link every point to the given scenario. Failing to link RNA polymerase activity to the specific experimental data provided in the question, that is a fatal error. Honestly, it doesn't matter how incredibly well you understand transcription. If you don't apply it to their specific context, you are going to lose those marks. Okay. Let's look at Section 2, Escalating Stakes, dealing with harder data and statistical analysis. We're moving away from descriptive biological concepts now and stepping into the higher-order domains, handling information and experimental skills. This is where the truly difficult marks are hiding. When you're analyzing complex transcription data, evaluative thinking is absolutely mandatory. You have to know exactly when to apply a t-test for comparing means between two groups, when to use chi-squared for categorical data, and when to bring in correlation tests to evaluate relationships between variables. Evaluative thinking means weighing the evidence quality, identifying the limitations, and literally justifying your conclusions mathematically. Furthermore, if you're given a practical investigation related to mRNA formation, examiners are hunting for a very specific sequence. You've got to logically select your apparatus, follow sequential instructions, make highly accurate observations, identify any anomalies in your data, and then draw evidence-based conclusions. It's a sequence for a reason. If you skip a step in your written design, you skip a mark. It's as simple as that. Let's move on to Section 3, the top 3 warnings from principal examiners. Having looked at the ideal framework, we really need to cover what you must absolutely avoid. I've scoured the principal examiner reports for this syllabus, and here are the top 3 fatal mistakes candidates consistently make. Mistake number 1, vague answers. Like we established right at the start of this explainer, examiners demand highly specific, precise details. When you discuss RNA polymerase, you have to detail the breaking of hydrogen bonds, the complementary base pairing, the formation of phosphodiester bonds. Whatever you do, do not leave the mechanism vague. Mistake number 2 is honestly one of the most frustrating things to see: failing to use precise units and terminology. Think about it. If you're calculating the rate of mRNA synthesis and you just casually drop the units, or if you use everyday colloquial language instead of established biological terms, the examiner is going to penalize you immediately. No second chances. And our final warning, mistake number 3. This reinforces our earlier lesson perfectly. Answers entirely devoid of the specific given context will immediately hit a mark ceiling. For example, if the question is about a specific bacterial strain adapting to heat, and your answer just blindly describes standard transcription without ever mentioning the heat adaptation, you will simply not access the higher mark bands. To make sure you don't completely freeze up under exam pressure, here's a quick hack. Build your own custom acronym for all the sequential factors affecting transcription. Try organizing the binding, the unwinding, the base pairing, and the rejoining into one memorable or, hey, even entirely ridiculous word. It'll actually help your brain instantly recall those procedural steps when the timer is ticking down. So, we've covered the framework, the statistics, and the examiner warnings. But here is a genuinely terrifying thought. What if your framework is absolutely perfect? What if your biological knowledge of RNA polymerase is flawless, you include the context beautifully, and you still drop a full mark band? Well, the scary truth is, misreading a single command word in the question prompt will completely derail your perfect answer and cost you the grade. You simply cannot afford to let a misunderstood command word destroy all your hard work. Check out our next explainer to master the command words that actually decide your mark band. But before you jump over there, ask yourself this. If the absolute highest marks in biology are consistently awarded for demonstrating the process rather than just regurgitating the final answer, is rote memorization finally dead? Thanks for joining, and I'll see you in the next explainer.